So I want you to imagine just for a second that your DNA is this massive, incredibly detailed library.
Right, a giant biological library.
Exactly. And for decades, when scientists were looking for the root causes of complex diseases, things like heart disease or schizophrenia, they would walk into this library and they'd look for spelling mistakes inside the books themselves.
Because the books, in this analogy, are our genes.
Right. They are the actual sections of DNA that code for the proteins building our bodies. And honestly, that logic makes perfect sense.
It does. I mean, if a book has a typo, the biological instructions are wrong.
Yeah, the protein gets built incorrectly and you get sick. But when scientists started doing these genome-wide association studies...
The GWA scans, yeah.
Right, where they scan the entire genetic code of tens of thousands of people, well, they discovered this completely shocking truth.
And it really was shocking to the field.
Yeah, over 93% of the genetic typos that cause our most devastating diseases, they aren't inside the books at all.
No, they're not. They are hiding out in the blank spaces between the books.
The blank spaces?
Yeah. The stuff we used to just dismiss as junk DNA.
Yeah.
Or, the dark matter of our genome.
So how is it even possible that a typo in a blank space can cause a disease like schizophrenia? That is the core mission of our deep dive today. We are unpacking a, frankly, groundbreaking paper from Nature Genetics that finally shines a light on this dark matter.
It's a fascinating study because what this research proves is that those spaces, well, they were never actually blank.
We just didn't have the glasses to read them.
Exactly. We lacked the right tools. And the breakthrough here really centers around an epigenomic mark. It's called H3K27AC.
Okay, let's break that down because epigenomic literally means like sitting on top of the genome, right?
Right on top of it, yeah. So inside our cells, your DNA is wrapped tightly around these spool-like proteins. They're called histones.
Like sled on a spool.
Exactly.
Right.
And H3K27A is a specific chemical tag, an acetyl group to be precise, that gets attached to these spools. You can think of it as a cellular neon highlighter. When researchers apply this highlighter to the genome, it lights up those non-coding regions.
The blank spaces.
Right. And it reveals that they're actually densely packed with what we call active regulatory elements, or AREs for short.
So they aren't just empty pages between the books. They are literally the control panels for the whole library.
That's a great way to put it. They are physical switches.
Yeah.
And we classify them mostly as promoters and enhancers.
Promoters being the ones right next to the gene.
Right. Promoters sit right next door to turn the gene on. But enhancers, they can be located incredibly far away on the DNA strand.
Wow, really? So how do they work if they're far away?
They literally loop back around in three-dimensional space to physically touch and flip the switch. So if you have a typo in one of these switches, the gene itself, the book, might be flawlessly written.
But because the switch is broken, the gene never turns on.
Exactly. Or it turns on at the completely wrong time.
Okay, so let's map out how researchers actually track these switches down, because it takes a massive amount of data layering. I like to picture this as like a biological crime scene investigation.
I love a good crime scene analogy.
Right. So if you're trying to track down a genetic culprit, that first tool, the GWS, the genome-wide scan, that gives you the general neighborhood where the crime happened.
It says the typo is somewhere on this specific city block.
Exactly. Then scientists use a massive database called GTEx. That's the Genotype Tissue Expression Project.
GTEx.
And GTEx is like checking the local power grid. It measures the RNA to see which houses in that neighborhood actually have their lights turned on.
But just checking the power grid doesn't tell you how the lights are controlled.
Right. And that's where the Nature Genetics paper comes in. By using that H3K27AC neon highlighter, they are walking into the specific house and finding the exact light switch on the wall.
And the sheer scale of their investigation is what makes this paper so unprecedented. To find those switches, the team generated 387 genome-wide profiles.
That is massive.
Huge. They mapped the DNA of 256 different individuals. But here's the key. They specifically took samples from 4 very distinct areas of the human body.
Right. The brain, specifically the prefrontal cortex, the left ventricle of the heart, the skeletal muscle, and the lung.
Exactly those 4.
But wait, I'm stuck on one thing here with the methodology. If they had access to the entire GTEx biobank, which has tissue samples from everywhere in the body, why limit this huge project to just those 4 organs? Doesn't that leave out things like the liver or kidneys?
It does, and it sounds limiting at first glance, but there's a really profound biological reason for choosing those specific 4.
Oh, really? What is it?
It takes us all the way back to human embryonic development. So when an embryo first forms, it's just this microscopic structure with 3 primary layers. We call them germ layers.
Okay, germ layers.
Yeah. You have the ectoderm, which eventually forms the brain and the entire nervous system.
Okay, so there's the brain.
Right. Then you have the mesoderm, and that builds the heart and the skeletal muscles. And finally, the endoderm, which goes on to form the lungs and internal organs. So by isolating the brain, heart, muscle, and lung, the researchers captured a complete cross-section of all 3 foundational origins of human life.
Oh, wow. So it gives them a baseline map of how development is controlled across entirely different families of cells.
Exactly. It's a developmental master key.
And the switchboard they uncovered with this master key is staggering. Across just those 4 tissues, they identified 282,000 distinct control switches.
It's a huge number.
But what really caught my eye was how specialized these switches are. Because, you might assume that a switch keeping a cell alive in the brain is the exact same switch keeping a cell alive in the heart, right? Like basic cellular housekeeping.
You would think so, but the data tells a completely different story. Out of those 282,000 switches, only 14% are fully shared across all 4 tissues.
Only 14%.
Yeah. And those shared ones are mostly those promoters we mentioned, the basic on-off buttons right next to the genes doing that basic survival housekeeping.
Which means a massive 62% of the switches are entirely tissue-specific.
Entirely. They only exist, or they're only chemically active, in one specific organ.
That is wild.
And the complexity of the organ fundamentally dictates the complexity of its wiring. For instance, the brain requires 86,000 unique switches to function.
86,000 just for the brain.
Right. And that vastly outnumbers the heart, which only operates on about 31,000 unique switches. The muscle has 37,000, and the lung has just 21,000.
So it's just a striking visualization of the sheer computational power needed to run a human prefrontal cortex.
Exactly.
Okay, so let's do a quick 5-minute recap here just to bullet point what we've mapped out.
Good idea.
So first, GWAS tells us disease typos are hiding in our DNA's blank spaces. Second, the H3K27AC highlighter proves these aren't blank spaces, they are 282,000 mapped control switches.
Right.
And third, the vast majority of these switches are custom built for specific organs. So a quick question for you listening, if you had to guess, which of those 4 tissues requires the most custom built switches to function?
Well, as we just spoiled, it's the brain with a massive 86,000 unique switches.
Exactly. But as we dive even deeper into this map, the data reveals something totally unexpected about biological sex.
Yeah, this was a really cool part of the paper. Because the GTEx database includes diverse samples from both males and females, the team could control for all sorts of variables.
And when they did, they found 2,436 sex-biased switches.
Right. Regulatory elements that are either significantly more active in men or significantly more active in women.
The distribution of those is so interesting to me. So the heart and the skeletal muscle, they each contained over 1,200 of these sex-biased switches. The lung only had 23, but the brain had 192.
Yeah.
So what are those 192 male and female biased switches in the brain actually doing? Is it just general brain function?
No. They are incredibly specialized. When they looked at the genes controlled by those 192 brain switches, they found pathways highly enriched for the sensory perception of smell.
The sense of smell, really?
Yes, smell. And they also found pathways controlling the endoplasmic reticulum's response to unfolded proteins.
Okay, endoplasmic reticulum. Let's translate that real quick. Basically, you can think of it as the cell's protein folding factory.
Right, because proteins need very specific 3D shapes to actually work.
Exactly. But sometimes they misfold, they get tangled up. So the unfolded protein response is basically the factory's emergency alarm system.
It stops production and clears out the trash before those tangled proteins become toxic and kill the cell.
So what you're saying is our DNA has specific switches for this cellular emergency alarm in the brain, and those switches are literally dialed differently depending on biological sex.
Precisely. Which actually brings us to the core mechanism of how any of these switches get dialed up or down in the first place.
Right.
This is where the paper introduces a really critical concept called the haQTL.
Okay, haQTL. That is some dense scientific jargon. Let's break that down into plain English.
You can take it piece by piece. So QTL stands for quantitative trait locus.
Which means?
In genetics, a QPL is just a specific location in your DNA where a single letter spelling difference exists between 2 people.
Oh, okay. So at, position 1 million on a chromosome, I might have the letter A, but you might have inherited the letter G.
Exactly. That's the QTL. Now, the HA attached to the front stands for histone acetylation.
Ah, which is our H3K27 IAC neon highlighter.
You got it. So an haQTL simply means that a single letter spelling difference in your DNA physically changes the amount of chemical activation on that specific switch.
Okay, wait. So the switch isn't just a binary on or off. It acts like a dimmer switch.
Yes, a dimmer switch is the perfect analogy.
So if I inherit an A in my DNA, my dimmer switch is turned all the way up to 100%, and the gene pumps out proteins. But if I inherit a G, the dimmer drops to 20%, and the gene barely functions.
That is exactly how it works. And the researchers found 130,000 of these genetic dimmer switches across those 4 tissues.
Wow, 130,000.
And to organize all of them, they categorized these dimmer switches into 3 distinct flavors of tissue specificity. I actually use a mental image of a house to make this clear.
Oh, I love a mental image. Okay, I'm picturing a house. Walk me through the flavors.
Okay, so flavor number 1 is the haQTL shared switch. Think of this as the main electrical breaker in your basement.
The main breaker.
The physical switch exists in every room of the house, and the genetic dimmer effect happens everywhere equally. If you have that G typo, the lights dim in the brain, the heart, the muscle, and the lung all at the exact same time.
Got it. That makes sense. What's flavor 2?
Flavor 2 is the haQTL specific switch. Think of this one as a smart bulb.
A smart bulb.
Yeah. The physical light fixture exists in every room, but the remote control of the genetic dimmer effect is programmed to only work in one specific room.
Oh, I see. So the exact same DNA typo dims the light in the heart, but does absolutely nothing if it's sitting in the lung.
Exactly. Now, flavor 3.
What's flavor 3?
This is the IR-specific switch. Imagine a highly custom, extremely expensive chandelier.
Fancy.
Very fancy. The switch itself and the genetic typo that dims it only exist in one highly specialized room and nowhere else in the entire house.
And let me guess, we see this massive amount of custom chandeliers, primarily in the brain.
You guessed it.
That is a brilliant way to conceptualize it. The breaker, the smart bulb, and the chandelier. And, if we think about the practical application of this, we have thousands of these dimmer switches dictating gene activity based on a single letter difference tuned to specific tissues.
It changes how you see the body.
Totally. So let's do another quick recap. We got 2,436 sex-biased switches affecting things like smell. We've defined haQTLs as genetic dimmer switches, and we have our 3 flavors of switches.
Right.
So question for you listening. Think about your own workout routine. You might separate cardio days for your heart from strength training days for your biceps. But biologically, because the heart and skeletal muscles share an embryonic origin, they share thousands of the exact same genetic dimmer switches.
It's true.
So how does that change the way you think about full body workouts? Your cardiovascular health and your bicep strength are quite literally wired to the same epigenetic switchboard.
It completely blurs the line between different physiological systems. And honestly, that blurring of lines is exactly what allowed the researchers to solve the ultimate GWA's mystery here.
Oh, let's dive into that. Because we need to connect this map to a human being actually getting sick.
Right, the disease connection.
The researchers took this massive catalog of dimmer switches and they overlaid it with genetic data from 55 different complex diseases and traits.
They were looking for intersections, places where the known genetic typo for a disease perfectly aligned with one of these newly discovered dimmer switches.
And the hierarchical evidence they found was just stunning. Some of it validated exactly what you'd expect, like cardiometabolic traits like coronary artery disease linked flawlessly to the custom switches in the heart.
Yep. And lung function traits heavily enriched in switches found in smooth muscle.
Which proves that respiratory health isn't just about lung tissue itself. It relies heavily on the smooth muscle that wraps around our airways.
But the profound revelation of the paper, the real aha moment, came when they mapped the genetic typos for psychiatric conditions, specifically schizophrenia and bipolar disorder.
Right. Because if I'm looking at schizophrenia, I would assume all the genetic typos point straight back to those 86,000 custom chandeliers in the brain.
And they certainly mapped heavily to brain and neuron switches. But they also mapped intensely to the switches controlling our blood and our immune systems.
Wait. Typos for schizophrenia are actively altering immune system switches.
Yes, and this is huge because for years, psychiatrists have proposed this clinical theory that immune dysfunction and neuroinflammation are core drivers of psychiatric disorders.
Wow.
But what this epigenomic map provides is hard physical evidence. The genetic variants we associate with schizophrenia are literally, physically, turning the dimmer switches up and down inside immune cells.
But wait, let me push back on that for a second. How do they know it's the exact same typo?
What do you mean?
Like, how do we know for sure that a disease typo found in a massive population study is the exact same typo controlling the dimmer switch in the immune cell? Couldn't they just be two different typos that happen to sit right next to each other on the DNA by coincidence?
That is the most rigorous hurdle in genetic research.
Yeah.
And it's exactly why they utilize an algorithm called COLOC. It stands for colocalization analysis.
Okay, COLOG.
Yeah, and it's a Bayesian statistical framework.
Okay, Bayesian math. Let's explain that because it's crucial here. So instead of thinking of it like a simple binary fingerprint scanner that either matches or it doesn't, think of Bayesian math like your phone's GPS trying to locate you.
Oh, that's a good analogy.
Right. When you first open the Maps app, the blue dot has this huge radius. That's your prior probability. But as your phone connects to one satellite and then another and then maybe a local Wi-Fi network, it updates the probability with every new piece of evidence.
It shrinks the blue circle.
Exactly. It shrinks it down until it pinpoints your exact location.
And that is precisely how COLOC works. It looks at the genetic typo causing the disease and the genetic typo controlling the dimmer switch. And it continually updates the probability that they are actually driving the exact same signal.
Fascinating.
It calculates what's called a PP4 score. If the score is high enough, well, the GPS has pinpointed the location. It mathematically proves that these aren't just two typos living next door to each other. They are the exact same snippet of DNA.
And using this Bayesian triangulation, they found 1,070 strictly colocalized events across those 55 diseases.
1,070 times where they proved it was the exact same typo.
So we can literally draw a direct line from a disease to a specific tissue to the exact dimmer switch causing the problem.
It's incredible.
Let's do another quick recap. We matched 55 diseases to specific tissue switches. We found unexpected links, like the immune system driving mental health. And we used COLOC's Bayesian math to prove the typos are identical. So, question for the audience. If a psychiatric condition like schizophrenia is physically driven by inflammatory immune switches in your DNA, how should that revolutionize the way we treat mental health?
It really demands that we look beyond just traditional neurotransmitters, We have to start addressing systemic inflammation.
And to see how this plays out clinically, let's look at how the researchers used schizophrenia as their primary case study, because they isolated 54 specific colocalized switches for schizophrenia.
And in doing so, they actually solved a massive dead end in the field.
Yeah, let's talk about that dead end. Because in traditional genome-wide studies, scientists had found a primary genetic suspect for schizophrenia. It was a specific typo named RS77831.
Right, that was the lead SNP.
But when they tested it in the lab against standard gene expression data, it did absolutely nothing. It looked like a total false lead. Why did it look like a false lead?
Well, it comes down to how we usually test tissues. Standard tests, they're known as bulk haQTLs, take a piece of tissue like a chunk of the brain, grind it up, and measure the average gene activity.
Okay, so it's like putting an entire fruit salad into a blender and then trying to taste an individual blueberry.
Exactly, it's all blended together.
Yeah.
And many genes only misbehave under highly specific conditions within a single type of cell. But because the H3K27A highlighter maps the physical dimmer switches rather than just the blended RNA fruit salad, it caught the biology in the act.
So what did they realize?
They realized that the suspect, RF77831, wasn't the actual culprit. The real causal typos were hiding right next to it, altering these epigenomic switches that the bulk sequencing completely missed.
Wow. But finding the broken switch is really only half the battle, right? You still need to know which light bulb it turns on. And since enhancers can be located super far away from the genes they control, how did they figure out the wiring?
For that, they used a final computational algorithm called gLink, which stands for genetic link.
Okay, gLink.
And gLink uses something called fine map EQTLs to draw a definitive, highly accurate wire from the newly discovered DNA switch straight to its target gene.
Okay, let's demystify fine mapped really quickly. So our DNA is an inherited single letter by single letter. It's passed down in chunks.
Right, blocks of DNA.
So fine mapping uses advanced statistics to sift through those chunks and isolate the single specific letter that is actually pulling the strings.
Perfect explanation. And when they applied the gLink algorithm to schizophrenia using these fine-mapped variants, it pinpointed a broken switch in the brain that was directly wired to two very specific genes.
KRO7 and NMRA1.
Yeah.
What are those two genes actually responsible for?
Well, KRO7 handles vesicle transport. You can think of it as the shipping and receiving department of a brain cell. It moves vital chemical cargo around.
Okay, and what about NMRL1?
That one handles the cellular response to oxidative stress and redox changes. Basically, it manages chemical damage.
Okay, so let's put that together. A typo in a blank space dims the switch. That dim switch messes up how brain cells move cargo and how they handle chemical stress. And that accumulated damage literally leads to schizophrenia.
It is an incredibly elegant and precise mechanism. But, and this is the crazy part, it doesn't stop at the brain.
Right. The gLink algorithm uncovered something truly paradigm-shifting about the rest of the body. They found 11 distinct schizophrenia regulatory circuits operating in the heart and skeletal muscle tissues.
Which sounds impossible at first.
Right. Why would a genetic switch for schizophrenia be operating in my bicep or my left ventricle?
It all comes down to vascular cells, the blood vessels that supply oxygen to all these tissues. Through gLink, they identified genes like WBP1L, which is a known marker for perivascular fibroblasts in the brain.
And those are the cells that wrap around and support our blood vessels.
Exactly.
Which means vascular homeostasis, the actual physical health of your blood vessels and how blood flows through them, is a hidden driver of psychiatric disorders.
Yes.
The genetic markers for schizophrenia are actively operating in the blood vessels throughout your entire body.
It strongly implies that psychiatry and cardiology are much, much closer cousins than we ever realized in modern medicine.
So our final mini summary here. Advanced algorithms like gLink let us connect non-coding switches to target genes. And we learned schizophrenia is driven by oxidative stress in the brain and blood vessel dysfunction everywhere else.
Great.
So my final question for you listening, if genetic markers for schizophrenia are actively operating in the blood vessels of your heart and muscles, can we still call it strictly a brain disease?
That's a huge question for the field to grapple with.
It really is. Let's bring this all together. At the start of our deep dive, we talked about the 93% of genetic typos hiding in the blank spaces of our DNA library.
The dark matter.
Right. And what this Nature Genetics paper proves is that those spaces are not blank. They are a highly complex, intricately wired switchboard. By using the H3K27 highlighter to illuminate the switchboard across the brain, heart, muscle, and lung, scientists can finally see how a single typo in a dark corner of our genome dims the lights on crucial biological processes.
And honestly, the consequence of that discovery offers a massive paradigm shift for the future of medicine.
How so?
Well, think about it. Right now, when we treat a disease, we usually give a drug that floods the whole body. It targets a specific protein or gene everywhere it exists.
And that is what causes massive systemic side effects.
Exactly. But remember our 3 flavors of switches: the main breaker, the smart bulb, and the custom chandelier.
The custom switches that only exist in one specific organ.
Yes. Our DNA's non-coding regions are absolutely full of those highly distinct tissue-specific switches. There are dimmer switches that only exist in the brain or only in the lung.
Right.
So what happens when we invent therapies that don't target the gene itself but target that specific local switch?
Oh, wow.
If we can fix the custom chandelier in the brain without accidentally turning off the lights in the heart or the lung, we might be looking at the end of systemic side effects.
The end of side effects by targeting the switches, not the bulbs. That is an incredible thought to end on. Thank you so much for joining us on this deep dive into the epigenomic map. To everyone listening, keep questioning the world around you, keep looking past the obvious, and remember, whether it's in a massive library, a crime scene, or your own DNA, the biggest discoveries often hide in the blank spaces.